Hello friends, I am Surekha Rode, Assistant Professor, Department of Botany, Rajasri Chhatrapati Shahu College, Kolhapur. Today we will going to see apomixis. Apomixis is derived from two Greek words. Apo means away from and mixed, the act of mixing. The term apomixis was first used by Inkler in 1906. Apomixis refers to the occurrence of an asexual reproductive process in the place of normal sexual process involving reduction, division, and fertilization. Apomixis refers to the development of seed without fertilization. In other words, it does not involve sexual fusion for development of seed. It differs from amphimixis, which involves union of male and female gametes for development of seed. The plant which shows apomixis is called as apomictic plant. Types of apomixis mainly recurrent apomixis and non-recurrent apomixis. Recurrent apomixis, it includes vegetative propagation and ogamospory. In ogamospory, embryo sac is diploid and may be arise from a cell of the archisporium or from some other cells of the nucellus. In a recurrent apomixis, both the egg cell and embryo are diploid and the embryo sac is developed from the megaspore mother cell. Non-recurrent apomixis. It is non-recurrent because cannot be repeated. In this type, haploid embryo sac develops after meiosis of megaspore mother cell and develops into embryo without fertilization. Recurrent apomixis is divided into mainly two groups: vegetative reproduction. In this, plant propagates from material other than the seeds. Second, ogamospory. Ogamospory is again divided into three types: adventive embryony, diplospory, and apospory. Vegetative reproduction. In this type of reproduction, the plants propagate by a part of their body other than the seed, like from bulbs, corms, suckers, etc. Such a type of reproduction always performed by the sporophytic parts of the plants, which are arise from either vegetative parts or from floral parts like floral buds. For example, formation of the bulbs in the agave or production of tubers in the potato. Ogamospory. Ogamospory, in this type, plants are propagated by seeds, but the embryo in such seeds is formed by some other process in which normal meiosis and syngamy has been eliminated on the basis of development of embryo from a cell of the unreduced female gametophyte or directly from diploid sporophytic cells of the ovule like nucellus or the integument. The ogamospory is further divided into different types: adventive embryony, diplospory, and apospory. Adventive embryony. In this type of development, the embryo is not formed from the cells of embryo sac, but it is formed from the cells of sporophytic tissues outside the embryo sac like nucellus or the integuments of the parent sporophyte and not from the cells of gametophyte. Sometimes it is also called as sporophytic budding. In such cases, gametophyte generation is completely eliminated, hence there is no alternation of generations. Such a type of embryony is reported in citrus species Mangifera indica. Second, diplospory. In this type, diploid embryo is formed from the megaspore mother cell without undergoing meiosis. Such type of development is reported in Taraxacum species. Apospory. It is the process of formation of female gametophyte without the reduction division of the megaspore mother cell. Rosenberg in 1907 reported apospory first time in the angiosperm, that is Eraceum species, a family of Asteraceae. In this type, megaspore mother cell goes through the usual meiotic divisions, but all four megaspores gradually degenerate. The chart shows diplospory, apospory, and adventive embryony. Causes of apomixis. Apomixis is controlled by specific genes. Most of the apomixis are of hybrid origin and also polyploid. The apomixis also controlled by recessive genes and hence it is useful in the breeding techniques. In some plants, the environmental conditions play a vital role in shifting of sexual mode of reproduction to apomixis. A species found in India may be apomictic, for example, Aponogeton, while some species found in South Africa, it may be amphimictic. Significance of apomixis. Apomixis offers the possibility to produce independent offsprings without any variation that occurs due to the segregation or recombination. Second, it is useful in the evolution, especially in case of facultative apomictic or groups of plants where sexual and apomictic members coexist. It plays a vital role in the evolution of the species. Third, as gametophyte apomixis concerns only the female side, the reduced pollen can be used to pollinate sexual relative to study the inheritance of the apomictic trait. These studies are mostly associated with aposporic plants. Fourth, as it is the phenomena of embryo formation without the act of fertilization, the genome of parent plant is preserved and it is in the offspring. If apomixis takes place in a haploid cell or tissues, the number of chromosomes is maintained as n, while if it takes place in diploid cell, the number of chromosomes will be maintained double, that is 2n. The occurrence of haploid parthenogenesis is also important in the genetical studies, especially in the breeding where true homozygous material to be obtained. Thank you.